All right, everyone. Thank you. Thank you so much for joining yet another session at LMHI 2021 Global Colloquium. I welcome you all and I extend a very warm welcome to all our speakers, to our Honorable Chairpersons, Honorable Dr. Ronko Itamura and Honorable Professor Aaron Tokalung. I would also love to extend a very warm welcome to our moderators, Dr. Yvonne Folk and MS Sandy Yu. The host nation for the session would be China. And now I would request our honorable moderator to please introduce the theme of the session. Thank you.
Thank you. Thank you, and I'll welcome all to this section. And the theme of this section is about basic, fundamental, and preclinical researches. And Aaron would be your chair today for this section. And first, we really lovely to see so many abstracts about the basic science of homeopathy. In homeopathy, clinical trials really cannot stand by itself, and we really need more and more fundamental research so that our clinical trial can have a really solid ground. And it would be our pleasure to see so many lecturers today. And I would give the time to Sandy. I will give this time to Sandy to introduce our speakers for today. Sandy, please. Yeah, they講你咪你個入 Zoom 啊 So would you please unmute Sandy, please?
Requesting the IT support to please unmute Honorable Moderator, Dr. MS Sandy Yu.
We cannot hear you, Sandy. Okay, never mind. I would do it as well. So our first speaker today is Dr. Subhakar from India, and his topic would be about preparation, standardization, and anti-plasmodial efficacy of novel malaria nodules. And Dr. Subhakar Carl comes from the Department of Zoology, and is the professor there. And the department is from Punjab University, India. And welcome, Dr. Subhakar, and I will give the stage to you. Mr. Subhakar?
I think I can't see Sukhvikor in the list of panelists. Please check whether she is waiting in the hall. Or we can start with Dr. Belare, and meanwhile, I'll just see.
Okay. All right, then let us go. Maybe we can swap a bit of our sequence, and let me welcome our second lecturer, Dr. Jayesh Bellary. And Dr. Jayesh Bellary comes from the Department of Chemical Engineering and Department of Bioscience, and Bioengineering and Wakafni Research Center for Bioengineering, Indian Institute of Technology, Bombay. And the topic for today would be the innovative process for making of homeopathic medicine. And we are looking forward to that. Welcome, Jayesh, please.
Thank you. Thank you very much. I will get my screen shared. I hope my screen is visible.
Yes, sir, it is.
Yes.
Okay, and it's in full screen. Okay, great. Thank you. So I'm delighted to be invited by LMHI 2021 to present our work on an innovative process for making homeopathic medicines. Thank you for that introduction. I'm Jayesh Bellare from the Indian Institute of Technology in Mumbai. And this work has been done in collaboration with the Central Council for Research in Homeopathy, Ayush Ministry, as well as the Indian Institute of Technology, Bombay, with colleagues there. In particular, Dr. Raj Manchanda has been supporting this work greatly, as also Professor Dr. Khurana, and the bench worker on this has been Mayur Tengire. So I thank them especially for the work that I will be presenting now on their behalf. In this present work, we are innovating a rather disruptive technology to manufacture and potentize homeopathic medicines. This move will significantly improve the acceptance of homeopathic medicines amongst those who are skeptical. We know that the extreme dilutions used in manufacturing of these medicines, although beneficial, are the root cause of disbelief by virtue of the high dilutions used. To address these shortcomings, we have restricted the high dilutions to lesser numbers, well within Avogadro's limit, while keeping the number of succussions the same as the existing traditional method. Therefore, we do not compromise on the basic potentizing features of homeopathic medicines. So the main objective of this work has been to validate the new and more scientific process to make homeopathic medicines, which have a higher number of succussions and a lesser number of dilutions. There are several advantages to this new process. The main advantage is that super Avogadro dilutions are not used, thereby removing doubts that have plagued scientific acceptance of the physico-chemical basis of the homeopathic medicines. So this method of preparation of homeopathic medicines addresses the most important point from a material science viewpoint, which is what is there in the medicine after the sequence of preparation as per traditional or the new method. So the main advantage of this is that super Avogadro dilutions are not used. There are several other advantages as well. Firstly, it gives a similarity in structure and is non-inferior to the traditional method of preparation. Secondly, it is superior in manufacture by virtue of the ease of preparation process. As I shall describe soon, the number of steps are reduced, and it has a good quality assurance, quality control by virtue of being able to measure and see the particles involved. The third key and important point is that this new method retains the principle of dynamization. Other major advantages in this method are that it imparts reproducibility and avoids batch-to-batch variations, as in the old process, which was rather long and multi-step. This one is shorter, fewer steps with lesser chances of any possible errors. Therefore, this new method will provide fast, easy, and cost-effective manufacturing of the medicines. What is the need to do this work? Well, the needs are several, and there are several advantages for this innovative process of potentization. Firstly, it retains the principle of dynamization. Secondly, we define a nomenclature that is exact and accurate, which specifies both the dilution levels as well as the level of succussion. It tells us exactly what the extent of apparent dilution is and what the total number of succussions are, and names them in a manner that is analogous to the current system of naming. I shall describe this naming system soon. Thirdly, it helps in standardization. By standardization of the process and improving the quality, which will ultimately help in regulatory compliance, this method should gain acceptance quickly. Fourthly, it simplifies the process. It makes the process of potentization easier and less time consuming. The proposed process will also make the entire process more cost effective by virtue of requiring less time and less number of steps without compromising on the level of succussion that is used. Fifthly, the scientific acceptance can improve. This new method will help get better, wider acceptance to homeopathic medicines in the scientific circles as well as in the lay circles, which are skeptical by virtue of having such a high dilution used in this system of medicine. This is a chart or a table showing the comparison between the existing old method and the new method. In the old method or the existing method, there are a large number of steps that are used. As you can see in this table, one goes step by step up to a fairly large number that goes even beyond what is shown in this table. There are a large number of dilutions involved, and there are succussions at every step. But in the new method, which is shown in the right column, the initial potency, right? The initial potency here, which in this example of 5C30C, indicates that the level of dilution is equivalent to the old 5C dilution. Whereas the next set of numbers, which is 30C, tells us that in this particular sample, the number of succussions used is equivalent to what in the traditional method amounts to the number of succussions given to a 30C sample. The number of dilutions, as you can see, is limited to 10C, which prevents us from diluting further than Avogadro dilution. And therefore, although in super Avogadro dilutions, 2 nanoparticles are seen, in this method, even theoretically, it is not crossing the threshold of super Avogadro limits, and therefore there can be no controversy with respect to the dilutions involved. Whereas, keeping the principle of dynamization through succussions intact, the number of succussions is given as per the old method, wherein we can have as many succussions as needed according to whether the number of succussions correspond to 10M or 1M or CM. So the method involves, the nomenclature involves 2 numbers. The 2 numbers clearly specify the extent of dilution and the extent to which succussion is given. So it's a completely defined nomenclature, and it shows how the process goes as well, which we shall elaborate in the next few slides. To explain further, what we have in this chart is to show typically the exact comparison between the old method and the new method. The first few steps are similar in that titration is done with lactose for any starting material. The next several steps are also similar in terms of having successive steps of dilution with a diluent or a solvent or a dispersant, typically 90% ethanol and water. In the new method, taking again the example of 5C30C, the extent of dilution used is exactly the same as 5C of the traditional sample method, which is 1:10⁻⁻10, and the number of succussions used is similar to the 30C sample, which corresponds to 300 strokes. So each of these nomenclated samples has a very precise dilution level and a succussion level, and it is possible to do this sequence of dilutions in far fewer steps than it is done with the traditional method. Here is an overview of the preparation process, and here we take the example of zincum met. So zincum metallicum homeopathic medicine manufactured by the new method is very analogous to the traditional method, the conventional method specified in most pharmacopoeia. To begin, the titration process begins with 100 grams of zinc powder mixed with 900 grams of lactose. Titration is done for several hours, typically 6, at the end of which a quality control step is done with respect to particle size, which should be less than 10 microns. And then the next step of titration is done. And similarly, several steps of titration is done. After reaching 4x, the titurations are transformed into the dispersing medium, the solvent or the dispersant 90% ethanol, and then it is subject to the number of succussion steps with dilution steps. So the dilution steps are 1:10, and the number of succussions typically, right? Typically, the number of succussions will be of the order of 100 per step or 10 per step, depending on what is specified. The automatic machine, which is machines which are used, and there are several available. Here is just one example, can do several thousand succussions in a day. And there are other machines which are faster or can handle much more sample than this one. These are standard methods of preparation. So there is no need to invest in new machinery to do this. It is simply a process change which can be easily adapted to current manufacturing lines. This is an overall graphical process of the method of manufacture as well as the quality control. The initial mixture of zinc powder with lactose is saturated as I described, followed by the ethanol water succussion. And then the quality control can be done in at least two ways. The simpler way is to have an electron microscopy characterization, which I shall be mentioning more about, where the nanoparticles can be seen of the original substance, in this case, the zinc. It can be analyzed for its structure and size. And this can convince regulatory authorities of the presence of the starting material in the final product. And it can be analyzed for composition by techniques such as energy dispersive X-ray analysis, which is a part of many electron microscopes today. Besides this, there are other spectrophotometric methods and spectroscopic methods which can be used in order to analyze these, particularly for metal-containing samples. In the electron microscope, one can get bright field, dark field, and diffraction patterns, which then allow us to very clearly demonstrate the presence of particles and crystallites in the sample. Straightforward to document these in a quality assurance manual and thereby show that there is no controversy with respect to the presence of an active ingredient in the medicine. The active ingredient present in the medicine could have modes of action that could be several, which are yet being researched. And the scientific session here and on subsequent days could throw more light on this. It will have effects with respect to directly acting on the cellular machinery, or it could have indirect effects through other means that are hypothesized and yet to be proven. What we have shown in our previous work is that there are these nanoparticles of the original substance which are formed thanks to the titration process and the succussion process. And during the succussion process, the alcohol water mixture leaches out components from the walls of the glass container, and those components then coat the particles. So we essentially have a system in which there is nanoparticles of the starting material coated with silica. And one can see that in the presence of these nanoparticles, they get coated with the silica chains. And there is an animation which may not run so smoothly online, but the result of the process is that we have these nanoparticles of the original substance, and in this case, this is natrium mur, which is coated by these silica chains to give us a controlled release particle of the sodium chloride, which otherwise would have dissolved away within seconds or minutes. So the main objective of this work, which I shall show in the next few slides, besides the innovative process, is to show equivalency with the old process. Equivalency has to be demonstrated in several ways. The first way is to show the morphological and the physico-chemical equivalency. Thereafter would come the biological and eventually the clinical, which are longer steps ahead. In this first step, we wanted to show the morphological and the physico-chemical equivalency of this process. What we have in this slide are the images. These are the images which show what the structure of these samples are as seen in bright field. That is, we see the particle shape. In the dark field, we see the internal crystallite shapes. And in the selected area electron diffraction, we can analyze the crystallographic nature of the samples present. And in the next column here, we have the EDS, which is the energy dispersive X-ray, which identifies the elements present in the sample. So we can see that these are the results of the high resolution TEM of zincum metallicum, which shows bright field, dark field, and the EDS patterns. The EDS clearly shows the prominent peaks of zinc present, some silica, sodium, and calcium present. There is copper also present, but this copper comes from the copper sample holder, which is typically used in electron microscopic analysis. The copper grid is coated with carbon in order to hold the sample, and that's why there is a carbon peak also seen. The bright and dark field images shows that the particles are surrounded by a silicate coating, and we can get the fingerprint of these particles by the electron diffraction patterns. Here are the results of the HRTM analysis of ferrum metallicum, which also shows similarly the bright field, dark field, selected area, and the electron diffraction images. From the electron diffraction, one can see prominent peaks of iron and silica, silicon present, silicon from the silica. The bright and dark field images should show particles surrounded by the silica coating, along with the fingerprint diffraction pattern, the selected area diffraction pattern, which tells us that the sample is indeed of iron, along with some silica present. Similar results are found for cuprum. In the case of cuprum, we have changed our support grid from copper, which would have interfered, to nickel grids. And here the copper signal that comes is from the sample itself. One can also now see a nickel peak, which comes from the sample holder. So the results of the cuprum metallicum tell us that the presence of copper is there within the sample, and the peaks of other ingredients present can also be explained. The nickel coming from the grid, the copper is the active ingredient, and the silica, oxygen, and calcium are from the glass wall, which has leached out into silicates. All these images I have shown you match very well with the traditional system of medicine, and a side-by-side comparison has been done to show that there is equivalency, complete equivalency between the nanoparticles in terms of their general shape and size, and in terms of the chemical constituents that form them. So here are the particles of cuprum met, which show again the presence of many of these system of medicine. The elemental mapping again shows the presence of these particles. Here we can in fact do the mapping. The crucial difference between this image and the earlier ones are that each of these images now show within one particle what is the distribution of copper present inside the particle, and it shows an abundance of the copper within the particle at the various in the various potencies of the medicine. So what we have done here in this study is to propose a method of preparation, and importantly to validate that method of preparation. So the new process has been validated as far as physicochemical and morphological equivalency is concerned. The new process has many advantages. It allows a validation exactly as I have shown today, which can be rather easily done for every batch of manufacture by every manufacturer using an outsourced facility or perhaps an in-house facility, much like electron microscopes, which were esoteric systems earlier, today are the standard part of every manufacturing line for a semiconductor manufacturing, microelectronics manufacturing plant. Similarly, I see that this would be a simple way of validating and bringing it into a quality assurance scheme to show that there is presence of a starting material within every medicine, every batch of medicine prepared. And therefore, this new process makes it easy for quality control as well as acceptance by many drug authorities, which are more general and becoming more integrated today. And there is a need to really demonstrate not only the mechanism of action, but the presence of an active material, which this method can easily show. We have shown this for metallic medicines such as cuprum met, zincum met, and ferrum met, which I've shown you in the images I've shown earlier. We've shown that the HRTM images, both bright field, dark field, the selected area diffraction pattern, and the electron dispersion X-ray patterns, as well as the scanning transmission electron microscopy, which shows the distribution of each of these elements within the nanoparticles, show that there is morphological and physicochemical equivalency within the system. This method of preparation is under patent filing, and a research article on this is in the process of is ready, and in the process after some revision is ready to be communicated, and we hope to do that soon. Thank you very much. I am happy to acknowledge my collaborators who are listed here, in particular Dr. Raj Manchanda, who's present, Dr. Anil Khurana, who's also present, Mayur and Professor Suresh, Bineet, and Ritika. Thank you very much, and I shall be happy to address any queries or questions now or later as the format allows. Thanks again.
Thank you for Dr. Kort, Dr. JS, wonderful presentation. And the next presentation will be presented by Dr. Subir Kort. Dr. Subir Kort, are you with us? Dr. Subir Kort, are you with us now?
Yes, we can see her here in the chat.
She's not responding.
Yeah, I'll just respond.
Okay. Thank you very much.
Good morning.
Good morning. And Dr. Subir Kort is from the Department of Zoology of the Pandit University from India. And the topic of today's presentation is preparation, standardization, and anti-plasmodial efficacy of novel malaria nodules. Let us welcome Dr. Kort.
Yeah, thank you for the introduction. And at the outset, I would like to thank the organizers for giving me this opportunity to share. There's some background. Sorry.
I would request everyone to please be on mute. Yes, please, ma'am, please.
So at the outset, I would like to thank the organizers for giving me this opportunity to share some of the wonderful work which we have done on homeopathy. And I would also like to thank the Central Council for Research in Homeopathy for making this possible, with the initial help from Dr. Manchanda, who was instrumental in starting this project, and later on with Dr. Khurana, who was also instrumental in helping us, continue this work. And then we have also filed a patent for this work. So I will I hope you can see the screen. My screen is visible?
Yes, ma'am, perfectly visible.
So the work I'm going to present today is on the preparation, standardization, and anti-plasmodial efficacy of a novel malaria nosode. So before going into further about the malaria nosode, I would just like to show that talk something about malaria. I think all of us are well aware about this deadly disease which is caused by this protozoan parasite Plasmodium. The disease is transmitted through the female Anopheles mosquitoes, and this is the parasite. I have shown the structure of the parasite, which has an apical complex through which it is able to invade the different cells in the host. Now, the disease is very deadly. All of us know the symptoms that are caused by this disease, and the life cycle also is well aware that the parasite enters in the stage of sporozoites, and then initially it enters the hepatocytes where it multiplies. So this cycle, we call it as exoerythrocytic cycle, and then once the merozoites they exit from these liver cells, they enter the red blood cells, and where again they go undergo the erythrocytic cycle, and then the parasite keeps on multiplying, and then we know the sequence further. They form the gametocytes, and then these are again picked up by the Anopheles mosquito, and stage when the parasite enters into the liver cells, as well as the especially the red blood cells, all the different types of clinical symptoms they are caused. Now, to see the global burden which has been shared by WHO in 2021, the malaria endemic zones with less than 10,000 cases has grown from 26 to in 2000 to 47 in 2020. And in the same period from 2015 to 2020, the number of countries also have increased, with the 10 indigenous cases have increased from 20 to 23. Now, so nearly half of the global population is at the risk of malaria. And if we see the status of malaria in India, it represents 3% of the global malaria burden. And the main states which are affected are Andhra Pradesh, Chhattisgarh, Gujarat, Jharkhand, Karnataka, Maharashtra, Odisha, Rajasthan, West Bengal, and Madhya Pradesh. Mostly Odisha and Madhya Pradesh are the one which are badly affected. And a recent survey has also shown that out of the 579 malaria patients that were studied in southwestern India, 62.9% they had Plasmodium vivax infection, 25.9% had Plasmodium falciparum infection, and 11.2% had mixed infection, which again is a very alarming signal, the rate at which these mixed infections are occurring. All of us are well aware that it is much easier to treat Plasmodium vivax infection, but when it comes to Plasmodium falciparum, it causes a very deadly disease, and most of the drugs which are available for treatment of malaria, we know that they are, slowly, they do have many side effects, but slowly most of these drugs are showing resistance. The parasite is showing resistance to most of these antimalarial drugs. And then a re-emergence of the disease is also occurring in most of these areas where sometimes you think the disease is eradicated, but slowly after some time you see that there is a re-emergence of this disease. So this urgently highlights that we need to do a consistent effort to develop new chemotherapeutic compounds, and also have look for some of the alternative therapies which could be used for treatment of this deadly disease, because we know that most of these allopathic drugs they do eliminate the parasite, but the major challenge is that they have lots of side effects, and another challenge is that resistance to these drugs has started appearing. So if we look for an alternative therapy, the best therapy which comes to our mind is the homeopathy, because it is a very natural, a safe drug which can be given orally, and most of the and it does not resistance to these drugs does not occur. Now, if it among these homeopathic drugs, I think since all the people, most of the people who are attending this conference, they are homeopaths, they are well aware about the advantages of a nasode and what a nasode is. So I will not go into much detail about the nasodes. So with this basic background, last 5 years back, I think we were given the responsibility by the CCRH to prepare these different types of nosodes from the parasitic material, and then MOU was signed, and under this work was carried out in collaboration with Central Council for Research in Homeopathy with Dr. Manchanda and Dr. Khurana and Dr. Naik, who was also instrumental in helping us do this work. So for this, when we planned to prepare different types of nosodes. We prepared cell-free parasite nosode, we prepared infected RBC nosode, and we also prepared a mixture of these two types of parasites. Then we potentized these nosodes and we checked the sterility. We also checked the in vitro antimalarial activity of these nosodes on the chloroquine sensitive strain as well as a chloroquine resistant strain of P. falciparum. We checked the in vitro cytotoxicity of these nosodes. We also checked the in vivo studies whereby we checked the suppressive activity of these three types of nosodes in the animal model, that is the mice. And then we also checked the biochemical estimations we did for liver and kidney function of the infected as well as the drug treated animals. We did histopathological studies to see whether they cause any toxicity in the infected and treated animals. We also did histopathology on the normal animals to see the toxicity caused by these nosodes. We also analyzed the cytokines to see what kind of immune response is generated. And out of these, the best nosode, we then did the preventive as well as the curative activity. To start with, first important thing was to maintain the Plasmodium falciparum culture in the laboratory. So we obtained these two strains, chloroquine sensitive strain and the drug resistant strain, from the National Institute of Malaria Research, New Delhi. And then we maintained the parasite in A+ human erythrocytes using RPMI 1640 by following the standard method of Traeger and Jensen. So we also used 15% inactivated human serum to grow this parasite. And after growing the parasite successfully in the laboratory, then we started the preparation of the nosodes. So the first nosode that was prepared was the cell-free parasite nosode, which we called it as N1. So for this, we cultured these parasites, and then after culturing, we counted these parasites. And after counting these parasites, then we lysed the RBCs to get the cell-free parasite. And then after centrifugation, we obtained the pellet. And then this was a method which was used for preparation of the nosode. And for preparation of this nosode, the basic material was given to this Schwabe laboratories for further preparing the 30 potency of this nosode. Now, the second nosode that was prepared was from the infected RBCs, and for this again we took both the strains of the parasites, drug resistant and the drug sensitive strain, and then we centrifuged this and obtained this pellet, and then this was then further used for the preparation of the nosode, where we used this first one part of the parasite pellet and 9 parts of the alcohol. Again, this part was not done by us. This was done by the standard laboratory. Which was suggested by the Central Council for Research in Homeopathy. Third, we thought that to improve the efficacy, maybe we could have a mixture nosode. We called it as N3, in which we put all the 4 types of parasites, that is the cell-free parasite as well as the infected RBCs, of both the strains, the chloroquine resistant strain as well as the chloroquine sensitive strain. So this, where 4 types of parasites were present, we called this as the N3 or the mixture nosode. Now, before going further with the studies, we wanted to check the sterility of these nosodes, for which we consulted the microbiology department in PGI, and we were suggested to use these different types of agar media, McConkey agar, blood agar, and SD agar, and we found that no growth was obtained in any of these different media when the nosode was, this streaking of the nosode was done on these different types of media. So which showed that there is no absence of any kind of contamination in the nosodes. Then we went on with studying the in vitro antimalarial efficacy, and which we do by the standard method of Schizont maturation inhibition assay, where we culture these parasites in the presence of different volumes of these nosodes. And this then we checked, and we found that when we add these nosodes to the growing parasite, then it inhibits the growth of the parasite, where the schizons are not able to mature and the merozoites do not come out. So that is the kind of assay we do, where this is the formula we use: number of schizons in the drug treated well by the number of schizons in control well into 100. So we found that 71.4% of the inhibition was obtained when the volume of this nosodes that was used was 160 microliter. And this was in the case of the N3 nosode, which was followed by the 62.85% inhibition when we used the N2 nosode, and 60.61% when we used the N1 nosode. So this, with the standard drug chloroquine which was used, we found the inhibition was 75.38%, which was quite good as compared to this N3 nosode. Inhibition was very good if you compare it with the standard drug chloroquine. Now, this was in the results when we used the resistant strain of the parasite, chloroquine resistant strain. So if you see, if we use the standard drug again, chloroquine resistant strain, the inhibition is only 5.26%. But when we used our mixture nosode, the inhibition was 68.57%. So this is a drug resistant parasite, and our nosode was able to inhibit this growth of the parasite to the tune of 68.57%, followed by 60% with N2 nosode. And so these results were very encouraging, because we found that these nosodes were also efficacious against the drug-resistant strain of the parasite. Then we checked the cytotoxicity on the standard methodology which we use in our laboratory on the raw macrophage cell line. And this we do through the MTT assay, and we found that all the nosodes, they were safe and non-toxic to the raw macrophage cell line, as the cell viability which was calculated was, you can see, more than 7% at all the tested volumes with all the different types of nosodes. Then we went on to do the in vivo studies, and where we found that we used different groups of animals. So we had the normal control, we had the infected control, which were infected with Plasmodium burgei. Then we used one group that is a vehicle control, who were given the potentized alcohol. Then one group was treated with N1 nosode, and then there was a one positive control. So this was the first set of experiments, and then we had a second set of experiments where we treated them with N2 and the N3 nosode. Now, what was the need for doing two different sets of experiments? So in the first set of experiment, we treated these animals with the N1 nosode, infected animals with N1 nosode for 7 days. And we found that the mice died after 19 days. So we thought that maybe the 7 days period was not sufficient for the treatment of these animals. So in the second set of experiments, we carried out the treatment for 15 days, and we found that there was complete clearance of the parasite. So we did not repeat the N1 group. So we repeated this with the N2 as well as the N3 group, where we had 6 animals per group, and we checked the parasitemia and different parameters on day 17, 14, 21, 28, and 35. So these are the groups of animals which we studied. I think I've already shown this. Now, we also gave the animals, the nosodes were diluted in the ratio of 1 to 10, and then they were administered for 50 days. Chloroquine was given at the rate of 10 milligram per kg body weight, and this was given only for 4 days, as is the standard treatment in the case of the chloroquine. Now, these are the results where you can see in the first day of first set of experiments when they were treated for 7 days, the parasitemia was in infected controls 32.264%, and the mean survival time of these animals was 9.33 plus minus 2.52 days. Whereas in the case of the chloroquine treatment in these animals, they had a survival for 25 plus minus 5 days. So chloroquine is when chloroquine is given, all the animals were able to survive. But in the case of N1 treated nosode, the animals did not survive for a much longer period of time. So in the second set of experiments, again, we treated these animals, and in the N1 treated, the animals died between 14 and 19 days with a parasitemia of 38 and 40%. Now, when they were treated with the N2 nosode, parasitemia was 28%, and by day 16, but then it increased to 34.25% in one of the animals. We also used the potentized alcohol as a control, and we found that all the animals, they died when they were treated with the potentized alcohol because of the parasitemia. So the best efficacy was observed in the infected animals when they were treated with N3 nosode. And on day 16, you can see the parasitemia was only 1.17%, and which further reduced to 0.005% by day 35, which showed that N3 nosode was the one which could actually eliminate the parasite from these animals, and then that is how these animals could survive for a much longer period. Most of the animals, they survived. Now, then we did the biochemical estimations, where we did study the ALP levels, SGOT, SGPT, and bilirubin. And if you see that wherever the parasitemia was there, the levels were raised, and as soon as the parasitemia was eliminated, all these biochemical parameters, they also started declining. You could see the increase in the levels of ALP in N1 treated animals, SGOT, SGPT. Most of these parameters were deranged, and whereas in the case of these kidney function tests, also you can see that some of the levels were normal, but later on some of the animals, some of the levels were not within the normal range. So whenever the parasitemia is more, the levels are deranged, and as soon as the levels of the parasitemia, they come to normal level, then these biochemical parameters, they are also within the normal range. So it is just to demonstrate that it is the parasite which causes derangement of these biochemical estimations, and it is not the nosodes which are having any kind of a pathogenic effect. But so this was further seen in the case of when the animals were treated with the N2 nosode, then and the N3 nosode, you can see that most of the parameters, they were coming to the normal level. You can see only these SGPT levels were normal, the bilirubin levels were normal, the SGOT was normal, and the ALP also was within the normal range, which shows that once the parasitemia is eliminated, the parasite is eliminated from the system, then slowly all the biochemical parameters, they also come within the normal range. So same was true for the kidney function test also. Then we also carried out the histopathological studies to see that whether it is only the biochemical parameters which are deranged, or also there are some changes in the histopathology. So this, some of the histopathological slides of the normal mice, the infected mice, and the drug-treated animals. This is of the potentized alcohol. So just to sum up, I will just show you that in the case of the animals who were treated with the N3 nosode, the most of the histopathology of the this liver, it showed that it was also almost the normal kind of histopathology was seen as compared to the infected animals, where a lot of hemozoin deposition, and there was chronic focal hepatitis, and so many different types of these inflammatory cells were seen. Now, in the case of the spleen also, by day 35, you could see that there is the non-follicular lymphoid hyperplasia, which was there in the infected animals, that was all over, and even hemozoin deposition, which was seen in earlier stages, that was also eliminated. So this was in the kidney also by day 35, the levels were normal. So this showed that then further, I told you that just to see whether the nosodes are causing any pathogenicity on the animals. So we carried out histopathology by treating these animals, normal animals, without any infection. They were treated with the nosodes, and you can see here that in the case of the liver, the sinusoids containing Kupffer cells, you can see they were all normal in appearance in all the groups of animals, whether they were treated with the alcohol, whether they were treated with N1 nosode, N2 nosode, and N3 nosode. Same way was for the kidney, they were treated with the alcohol, N1, N2, and N3 nosode. So all were normal. The normal morphology of the kidney was seen, and same was true for the spleen. So spleen also, this is alcohol, N1, N2, and N3 treated and nosode animals. So this showed that these nosodes, they do not cause any kind of biochemical as well as histopathological changes in the animals, and whatever biochemical changes that were seen in the infected animals which were treated after nosode was basically because of the parasite which was persisting in the cells, and because of that all these changes they were seen. So further we wanted to see the cytokines on the different types, on the different days of treatment, and we found that we tested these cytokine levels, IL-4, IL-10, IL-17, and gamma interferon by using the commercial cytokine ELISA kits. And we found that high levels of these cytokines were seen. You can see until you can see these are the different groups of animals. Is the normal control, infected control, animals treated with alcohol, and this is in the N1 treated animals. So initially these IL-4 levels as well as the IL-10 levels, they were increased, and but after treatment with the, I will show in the next one, when they were treated with the N2 and N3 nosode. So this was results of the first experiment, and N1 experiment where they were treated only for 7 days. And most of these cytokines, we know they also play a significant role in the pathogenesis, in causing the pathogenicity which is caused due to the these parasites. So most of these cytokines, they were increased during this treatment. Now, in the case of the interesting results were obtained when the animals were treated with N3 nosode, where you can see that the levels of anti-inflammatory cytokines IL-4 and IL-10, they were observed to be increased, whereas there is decline in the pro-inflammatory cytokines, interleukin-17 and gamma interferon. So this shows that when these pro-inflammatory cytokines are decreased, this also correlates well with the elimination of the parasite from the system. So once we found that N3 is the nosode which is showing very good efficacy as compared to the N1 and N2, we further carried out the preventive studies, and in the preventive studies, the treatment of these nosodes is done on day 1 and continued for 15 days. And after that, on day 16, the animals are infected with the Plasmodium burgeri infection. So after 7 days of this infection, then slides or blood smears are made to check the percentage parasitemia, and we found that the mixture nosode, that is the 33 potency which we use for treatment, a maximum parasitemia of 4.14% was observed, and all the mice in the uncontrolled died by day 8 inoculation, whereas the others, they survived. Now, then we did these liver function and kidney function tests of these animals, and we found that only other animals, they were dead. Only 4 animals, they survived, and we found on day 21 that you can see the levels of these biochemical estimations, that in the animals who survived, the levels were almost in the normal range, apart from the increased SGPT levels. Now, same way, the kidney parameters also were slightly increased in the case of these animals on day 14, but you see by day 30, they were in the normal range. So this demonstrated that the mixture nosode, it exhibited promising antimalarial activity against both the Plasmodium falciparum in the in vitro system and the Plasmodium burgeri system, which we use for the in vivo drug efficacy studies. By the biochemical and histopathological studies, we also highlighted the safety of the nosodes for the rodent host, and the study provides valuable insight in the novel medicament that has potential to be used for the treatment of malaria. Now, these nosodes, they are prepared from the protozoan of genus Plasmodium, and out of these nosodes, we concluded that N3 was the best nosode, as it had in vitro antimalarial activity. Also, it showed a very good in vivo antimalarial activity against the Plasmodium burgeri infection. It also showed safety. In addition to that, it also showed that the levels of anti-inflammatory cytokines, which are increased, and while decline in pro-inflammatory cytokines was evident, which showed the clearance of parasites. So this is additional, proof that the parasite gets cleared because of the generation of this immune response, and it can be further used in the treatment of malaria after, doing further studies, and for which we have already submitted a further proposal to the CCRH, and let us see the fate of that proposal. So this work we have published in the Homeopathy Journal. Recently, it has come online, and I would again acknowledge the help rendered by Dr. Manchanda, Dr. Khurana, Dr. Nayak, for extending this project to us, and for which we are very happy that the first time we did this homeopathy work, and we came up with good publication, and in collaboration with CCRH, we have also filed an international patent. And then I would also like to acknowledge again Dr. Upma Bagai, who was instrumental in starting this homeopathic work on malaria, and then this work was extended. She left us 3 years back due to her sudden demise, and then after that, this work was taken over by me. And I would I never miss mentioning her name, because she was a very ardent follower of homeopathy, and she was really instrumental in starting this work in parasitology or homeopathic medicines, and which we were able to continue. And I should not forget to thank the students who are instrumental in doing this work, Mansi Suri and Sapna, along with Dr. Neha and Dr. Neha Walter and Varun, who carried out the in vitro work. All the in vivo work was carried out by Mansi Suri and Sapna. And thank you so much for patient hearing.
Thank you for Dr. Koff. For the next section, the presentation will be from Professor Wolkoff, and the topic will be the biological and physical chemical property of ultra-high dilutions of hydrated furans C60, prospective homeopathic remedies. Let's welcome Professor Wolkoff.
Good morning. Do you see my presentation?
Yes, sir.
Do you hear me?
Perfectly audible, sir.
Okay, that's nice. So first, of course, dear colleagues, it was an honor for me to give a presentation on this exceptionally important meeting. And I'm very thankful to the organizers of the Global Colloquium, and especially to Professor Raj Manchanda, who invited me to participate in this event. I'd like to report about our work, about our studies of biological and physical chemical properties of ultra-high dilutions of hydrated fullerene C60, which we suppose may be looked upon as a prospective homeopathic remedy. Here is the team, our team with my colleagues. We studied, worked on this problem for several years, and it was performed at the Moscow State University and also in the Institute of Biochemical Physics of Russian Academy of Sciences. First, I would like to remind you about what fullerenes are. Fullerenes were synthesized at the end of the 20th century in 1980s, and for these unique molecules, the Nobel Prize was given. They are unique because they represent a closed spherical structures, polyhedron, which are built from 3 coordinated carbon atoms. There are many different fullerenes, but most stable and found in nature representative of fullerenes is fullerene C60, which consists of 60 carbon atoms. It is built of 12 pentagons and 20 hexagons, and is the molecular object with the highest symmetry known. C60 represents a certain degree of aromatic character because of the 3 coordination of carbon atoms. Native fullerenes are insoluble in water, and that's why they are not easy to be used in biomedical studies. However, they may be dissolved in water either using solubilizers or after chemical modification. The most advanced method of their solubilization was developed in 1995 by the Ukrainian physical chemist Grigory Andreevsky. It allowed to enclose a native fullerene, which is colored here as blue points, inside a water cage, the water cluster of 80 water molecules, without the use of toxic solvents such as different solubilizers, or without its chemical modification. Here is the model of this hydrated fullerene, which was built by the British physical chemist Martin Chaplin. Such particle, the fullerene enclosed in water shell, was named hydrated fullerene C60, abbreviated here as HYFN. Solutions of hydrated fullerene C60 represent a hybrid of true and colloid solutions because hydrated fullerene is able to attract thousands of layers of water molecules, covering them into specific structured water, which has peculiar physical and chemical properties. As a matter of fact, it is not always reminded that many nanoparticles now, which are used in technique in biology and medicine, in aqueous systems attract thousands of layers of water molecules and modify the properties of this water. Fullerene, as it is, it behaves both as an antioxidant and it even sometimes is called free radical sponge, but also as a pro-oxidant, generator of superoxide anion radical and of singlet oxygen. Here is a sketch of how the process of action of fullerenes on molecules, on gas molecules may occur. Here is the fullerene molecular covered with water shells, and what is interesting that fullerene itself, hydrated fullerene, is negatively charged. That means that water shell around it is negatively charged. It has reducing processes. So on the boundary between bulk water and this water shell covering fullerene, the processes in which free radical reactions may occur. And in this sense, hydrated fullerene may serve as an organizer with processes with reactive oxygen or reactive carbon, reactive nitrogen species, and it may modulate free radical reactions which continuously, as a matter of fact, occur in aqueous systems. Fullerenes, there is a lot of reports in the literature about biological activities of solubilized fullerenes, of modified hydroxylated and other chemically modified fullerenes, and also of hydrated fullerene, of which we are talking here. In numerous publications, antibacterial, hepatoprotective, neuroprotective, immunostimulating, anti-atherosclerotic, radioprotector, anti-cancer, and even anti-aging activity of solubilized fullerene C60, and in particular of hydrated fullerene, was reported. Here, we questioned if hydrated fullerene could act in on different biological test systems in ultra-high dilutions, and if ultra-high dilutions of hydrated fullerene had peculiar physical chemical properties which differ them from solvent water used for their preparation. We prepared ultra-high diluted hydrated fullerenes by the traditional procedure from the original stock solutions of hydrated fullerenes using successive centisimul dilutions down to calculated, of course, concentration 10 to the minus 31 1 molar, in order just to overcome the so-called Arrhenius barrier. And with succussion, or better to say, the vortexing, prolonged vortexing at each step. So first, I would like to tell about our studies of the hydrated fullerenes of different ultra-high dilutions of hydrated fullerenes on different biological test systems. And in particular, we studied the effect of hydrated fullerene on the stability of enzymes which lose this stability either by prolonged incubation at room temperature or by heat desactivation. Here is effect of hydrated fullerene on bacterial luciferase activity, which was stored in solution at room temperature for several days. So here is the control activity of this enzyme, which, as you see, deteriorated for several days. And here is the activity of the enzyme to which hydrated fullerenes in different ultra-high dilutions was added. And one can see that hydrated fullerene in ultra-high dilutions, which are equivalent to concentrations down to minus 23 molar, are stored. So it stabilizes this luciferase against loss of activity. In fact, it worked on two different other enzymes which belong to different classes of enzyme activity. For example, here is the illustration of effect of hydrated fullerene with in moderate ultra-high concentration on heat-treated alkaline phosphatase. So phosphatase was heated for 10 minutes at 70 degrees, and it dramatically lost its activity. If fullerene were added to the alkaline phosphatase after heating, then surprisingly, there was a recovery, or partially revived heat inactivated enzyme. And if it was added before heating, also the activity of the enzyme was twice as high as in the heated control. So it protected in such a way from heat inactivation peroxidase, alkaline phosphatase, and luciferase from heat inactivation. Then we started, so these results are based on numerous experimental experiments which were performed with a lot of different controls. And here these differences between fullerene action and effect on enzymes is statistically very highly significant. We studied also effect of fullerenes on the energetic, bioenergetic processes which proceed in fresh, non-diluted human blood. We long ago found that if you take non-diluted human blood and add the probe for production of superoxide anion radical in this blood substance, which is called lucigenin, then one can see that there is photon emission from blood, which indicates of lucigenin-dependent photon emission, indicates of permanent production of superoxide anion radical in it. And hydroxyfullerene in ultra-high dilution and in normal dilution modulates redox processes, respiration in blood. Here is the intensity of photon emission from blood, from control blood, from blood to which 10 to the minus 6, 10 to the minus 7 molar fullerene was added, hydrated fullerene was added. Then, as could be expected, normally the effect disappeared, but when we went down to 10 to the minus 17, 10 to the minus 19 molar, then again, there was a very strong increase in photon emission. So fullerenes intensified processes the production of superoxide anion radical, which normally goes in blood of healthy human donors. Here are the data given for one and the same donor on two different experiments, which one was performed in July and another in September. But in fact, this data is characteristic for many different donors with healthy donors. So besides this, we found that hydrated fullerene at this ultra-high dilution attenuated intensity of luminol-dependent chemiluminescence of whole human blood, in which respiratory burst was induced, and also it inhibited cell proliferation and accelerated the process of aging of transformed Chinese hamster cells. We stress here that this line of Chinese hamster cells is malignant in cell culture. Moreover, secondary addition of this ultra-high dilution to cells that reach a stationary phase of growth caused a rapid death of cell population. So, given that C60 is not very active chemically, and that in hydrated fullerene, the C60 molecule is surrounded by water shell, one could suggest that its activity is due to unique properties of water shells formed around C60, which somehow changes the properties of all water solution containing ultra-high dilution of hydrated fullerenes. And that the effects of hydrated fullerenes in ultra-high dilutions are determined by some special features of aqueous systems that originate in the cause of its dilution process. Therefore, we compared some physical chemical properties of ultra-high dilutions in dilutions equivalent to concentrated, calculated concentrations down to 10 to the minus 31 molar, and corresponding dilutions of water prepared in the same manner. We studied different physical chemical properties of these ultra-high dilutions, but I'll concentrate on two kinds of the experiments. We titrated ultra-high dilutions of hydrated fullerenes with acid and then with alkali. So when we studied titrated it with hydrochloric acid, then it turned out that ultra-high dilutions of hydrated fullerenes in all range from 10 to the minus 7 to 10 to the minus 31 has higher buffering capacity against acidification than water dilutions, which is meaning that supposedly there are some species in their ultra-high dilutions which neutralize added protons to the system. Another interesting phenomenon which was found by us, that when, for example, you take some already acidified, especially in ultra-high range dilutions of hydrated fullerenes, then consecutive dilutions of either hydrated fullerenes or control water differ in their buffering capacity against acidification, especially in ultra-high dilution range. For example, here is the pH of different dilutions, and one can see that in acidified ultra-high dilutions, there is a very high difference between consecutive dilutions. For example, 10 to the minus 17 molar dilution has such pH, and next 10 to the minus 19 molar dilution has higher range, and so on. As a matter of fact, in water dilutions also there are such variations between the consecutive dilutions, but they are much smaller, especially in the high dilution range. Titration of hydrated fullerene dilutions and respective water dilutions with sodium hydroxide, the alkalization, did not reveal difference in their buffering capacity. However, consecutive dilutions of hydrated fullerenes differ in their buffering capacity, especially in the ultra-high dilution region, even much stronger than in experiments with sodium with acid titration. The range of pH fluctuations between successive dilutions is much more intense, especially as can be seen here in ultra-high region. So, and these differences are very highly statistically significant. As a matter of fact, in alkalized ultra-high dilutions of water control, there is no such variations of pH between successive dilutions. There was another test of the properties of hydrated fullerenes, which I'd like to tell the results of this test. That is dry droplet microscopy for the evaluation of the structuredness of ultra-high dilutions of hydrated fullerene solutions and water controls. The described above features of these dilutions of hydrated fullerenes and of the respective water controls may reflect the complexity of the structural properties of aqueous systems. And such complexity of structuredness can arise in the course of dilutions with intensive agitation of at each stage. And in order to find out if such heterogeneity of structuredness is characteristic for the studied preparations, we use the method of drying droplets of either hydrated fullerene dilutions or respective water controls to which microspheres that are water hydrophilic spheres with the diameters about 1-2 microns were previously added. So we added microspheres to different ultra-high dilutions of either hydrated fullerenes or control water, and then took droplets from these suspensions and dried out these droplets on glasses. This method allows to assess the degree of aqueous systems heterogeneity or structuredness. Here I present only a couple of examples, especially for ultra high dilutions, for hydrated fullerene preparations which are equivalent to dilutions 10 to the minus 31 molar and respective water control. And one can see that on the photos of these dried out droplets containing microspheres, which were prepared on this ultra-high dilution of fullerene, there appear these different figures So there is some kind of organization. Here are examples of 2 droplets, and there are much more, of course, droplets which we prepared in comparison to their respective water control, which are seem to be much less organized. And on the next figure, we can compare the number of drops with a substantially uneven distribution, the appearance of regular structures of microspheres in hydrated fullerene dilutions in comparison to water dilutions. So here we compare the appearance of droplets with heterogeneous microsphere distribution. For 10 to the minus 7, there is practically no such droplets, but already for 10 to the minus 9, there appear in hydrated fullerene ultra-high dilutions much more droplets with kind of organization, and with the higher and higher is the degree of dilution, the more and more organized structures can be seen in dried droplets prepared on hydrated fullerenes, and they appear also in patentized water, but in significantly less quantities. Hydrated fullerene dilutions are significantly more structured than the respective water dilutions, especially in the range of ultra-high dilutions. So we come now to the conclusions of the results reported in my presentation. Hydrated fullerene C60 in ultra-high dilutions exhibits a wide range of biological activity, expressed mainly in beneficial effects on variety of biological test systems. It stabilizes enzymes belonging to different classes against thermal and oxidative inactivation, and it is able to revive partially inactivated enzymes. Hydroxyfullerene in ultra-high dilutions modulate respiratory processes in whole human blood. Hydrated fullerene in ultra-high dilutions exerts cytostatic action on malignant transformed cultured cells. We suggested that biological activities of hydrated fullerenes is determined by special features of these aqueous systems. And indeed, we found that they had significantly higher buffering capacity against acidification than controlled water dilution. These results indicated of a higher electron donor capacity of this solution than respective water dilutions. Consecutive dilutions of hydrated fullerenes after either acidification or alkalinization differed in their buffering capacity. Most prominent differences were observed between consecutive alkalized dilutions in the highest range of dilutions, while no significant differences in pH between equivalent dilutions of control water were observed. And finally, basing on our own results and literature data, we assume that numerous effects of hydrated fullerene in ultra-high dilution in a wide range on a wide range of processes going in both in vivo and in vitro may be related first to their ability to make an aqueous environment in which these processes go on more ordered due to exceptional symmetry of water agonizer fullerene C60, and to the ability of the hydration shell of fullerene possessing reducing properties to modulate redox reactions with reactive oxygen species participation that continuously proceed in aqueous systems and perform important bioregulatory function. So due to all these features of hydrated fullerene in ultra-high dilutions, they may be considered as prospective entities for their application as homeopathic remedy with a wide spectrum of activities. And I would like just to add only that already there is in Russia water on sale which contains hydrated fullerene dilutions in ultra high dilutions, and this water is on sale, and in some medical there are some clinical studies not related with our group which show the beneficial effect of such drinking water. Here is the list of references of papers which were published on this topic. And I would like to end my report thanking again very much you for your attention. And again, I would like to thank Professor Raj Manchanda. Thank you very much. That's all. I finished.
Thank you for Professor Wukov. Wonderful sharing to us. Here come to the last section of the last presentation of our section. The topic will be the biological activity and physical property. The topic will be the biological activity and the physical properties of potentiate solutions by Professor Maria Tombridge. She's come from Faculty of Physics, Lomonosov, Moscow State University, Russia. Welcome, Professor Tombridge.
Thank you very much. Dear friends, dear colleagues, I'm very thankful to the organizers of this great event. And dear friends, dear colleagues, I'm very thankful to the organizers and especially to Dr. Manchanda, to the organizers of this great event. And I'm happy to join my colleagues all over the world. Thank you very much. Dear colleagues, mechanism of action for homeopathic remedies and their manufacturing is studying intensively all over the world.
I'm sorry to interrupt, but you are not yet in full screen. Would you place the slide? Yeah, now we are seeing your PowerPoint, but not the full screen. Or maybe you need to unshare and then share again, but then choose share your screen instead of sharing the PowerPoint apps.
I don't know why.
What you can do, ma'am, is at the very top of the screen, at the very top, on the title bar of the screen, there's a little button. There's a little button at the, yes.
Don't.
I believe you shall reshare your screen, as I think you only have shared this window with us, so it is not able to play a slideshow. Could you please try resharing it with us, ma'am?
Ma'am, Justin, can you go on the top? There are presenter viewer, please uncheck.
I don't understand. You can see my presentation. I don't know what to do.
You first unshare, so you stop the sharing.
Марина Суреновна, вы меня слышите?
Да, конечно.
Да, переключитесь, пожалуйста, на полный экран. Вот,
я переключилась.
Мы видим только стандартный экран и сбоку много слайдов. Ну, в принципе, на самом деле можно так продолжать. Если, чтобы не терять времени, все равно видно, основной слайд виден, можно так продолжать. Colleagues, I suppose that we should probably continue because the major slide is seen.
Right, all right.
So, yes, the major slide is seen. It's rather large dimensions.
Ну, сейчас не видно.
Вернитесь снова назад и я думаю, что имеет
Я
смысл.
вернулась назад. Давно.
Нет, сейчас мы видим последний слайд.
Ну, я не знаю, почему.
На экране последний слайд. Значит, слева у вас слайды.
Слушайте, я умею показывать. У меня на экране первый слайд.
Первый слайд, да? Ну вот что-то сбой.
I don't understand.
Сейчас мы видим третий.
I believe we can continue in the same way because majority of the slides are visible to everyone.
Сейчас первый слайд. Все. Сейчас первый слайд. Окей.
Is it okay now?
Just allow our IT support team a moment. They'll fix it up.
Is it okay?
No, ma'am. Can you tag in your sharing, ma'am?
Can you please close the screen share, ma'am, and then try sharing again?
Okay.
And this time when you share, when you click on share screen at the very bottom of the screen, you can see a pop-up. There you see screen written at the very first place in the top left-hand corner. Are you able to see that?
Я им дала право управления.
Just click on that.
Okay.
And click on share. Yes. Now, when you go to the very top, okay.
Can I continue?
I believe, yes.
У нас второй слайд уже, так что можно попробуйте продолжать, наверное.
Oh,
It's quite legible. Yeah, why don't we just go ahead?
Yes, yes. Please, 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 ma'am, continue.
okay. Okay, please. Conduct my demonstration. Okay, at the beginning of my report, I should like to make a brief review of the Russian studies of low and ultra-low concentration actions on biological models. And this research has really a great history. It seems to me especially important for homeopathy, studying effects using various biological and biochemical models. The meaning, the meaning of these models is that they are simple, unlike a living multicellular organism in which many complex regulatory reactions occur simultaneously. I would especially like to underline two Russian researchers' contribution to the understanding of the effects of low doses of potentiated and non-potentiated substances. For example, using above-mentioned models in 1986, Professor Burlakova showed non-monotonic effects of low and ultra-low concentration of various substances. 45 years ago, Professor Yamskova isolated substances from a living organism, substances that had effects in low doses, and justified their low concentration and dose action. A group of Russian toxicologists have shown that the effect of low and ultra-low doses was manifested itself in the presence of large doses action. This effect can be used and used already for detoxification. At that time,
Sorry, ma'am. Ma'am, I think your slide not moving, ma'am.
At that time, we conducted some experiments together with Professor Burlakova. For example, we demonstrated the influence of a new antioxidant substance, phenozan, in dilution of D25 on the viscosity of the cell membrane modification. Also, together
Ma'am,
with...
sorry, your slide is not moved, ma'am.
Ma'am, we believe there is a problem with the presentation. I think, can you please close the presentation and then restart the presentation, or else instead of using the arrow key, you can just mouse point and click on that. So
Now
that,
it's moving.
Yes,
yes, now
Now
it's
it's
moving.
moving.
Correct. Yes, sir.
please
Now it's moving.
conduct.
Now it's moving. So let her continue.
Yes, please.
Please continue, Maria. Now it's moving.
Сейчас слайды стали двигаться. До этого они все время был на втором слайде.
Оставь так, не переключай.
Я буду так показывать. Окей. Also, together with Professor Vladimirskaya, we observed effect of some homeopathic remedies, for example, aurum, lycopodium, arsenicum in dilution D30, and G, KSF, colony stimulating factor, on granulocyte macrophage progenitor cells from oncological patients. And we can see the action of these remedies, good action, normalizing action on oncological patients' cells, granulocyte macrophage progenitor cells. Together with Professor Kaminka, we studied the influence of some homeopathic remedies on the contractility of an isolated intestine. We can see in these experiments best spasmolytic effect for Hecla lava. And now, but in spite of clinical success of homeopathy and evidence-based biological investigations, there are limited quantity of physical arguments. And this is very important for the protection and promotion of homeopathy. Besides, we have not seen any work on the study of a number of serial dilutions, and it remains unclear how and why homeopathists choose any dilutions. In this aspect, since 2000, we have been conducting a number of experiments with serial dilutions of potentiated water and some potentiated substances. This report is devoted to the comparing of physical and biological effects received with serial dilutions of sodium chloride, sucrose, and potentiated water. The aims of the present study were to establish the correlation between physical properties of potentiated solution and their biological activities. To prove biological activity of potentiated water and to prove reproducibility of the obtained phenomena. Fluorescence, conductivity, optical microscopy, and weighting methods were used. Spontaneous motility of unicellular freshwater hydrobionts Infusoria spirostomum ambiguum and germination of seeds of Amaranthus albus were used as biological models. Germination was observed in disposable small plastic petri dishes containing 30 seeds and 2 milliliters of the investigated solutions. The main part of the experiment of both experiments was done using serial dilutions from 1D till 30D or from 1C till 30C dilutions of sodium chloride and sucrose. We have such reasons for this choice. Infusoria is a freshwater organism, and salt solution is a shock for this organism. The same shock influence has sucrose for plants for seed germination. We know that water and aqueous solutions are non-equilibrium, have immanent property of nanostructure self-organization, sensitive to weak chemical and physical forces. In this slide, we can see non-monotonous luminescence intensity at 345 nanometer for the potentiated solutions of sodium chloride in decimal dilutions from D1 till D13. The same results were repeated on the first day of preparing preparat, in 1 week, 2 weeks, in 6 weeks after preparation by Veleda in Russia. The same results was received at Unda manufacturer and remedy was done by Unda. We see exact reproducibility of our data. Changes in luminescence were compared with motility of spirostomes. Here we can compare the intensity of luminescence and motility of our Infusoria spirostomum. We can see the correspondence of the most intense luminescence of sodium chloride in D2 and D13, D14. No motility at all. In other words, we can see the correspondence of physical properties and biological activity. All these results were statistically significant at confidence level less than 5%. Detection of some active sodium chloride dilutions using nanoparticle tracking analysis Malvern demonstrated the presence of particles in all samples, but the most quantity and the largest size of the particle are corresponding the most active dilutions. Particles, and their mean. Next slide can demonstrates us these nanoparticles in reality. At this slide, we can see changes of fluorescence in potentiated water. In potentiated water luminescence, we can see rather great changes, and we can observe them also during all our experiment, during 6 weeks. Now, these results, these results can assume us that the process of potentiation itself causes changes in properties, not only with substance, but without any substance. These results were reported on SPIE Congress in USA. Similar patterns regarding what we saw earlier for germination. Germination in cases of potentiated sucrose stops under the influence of dilution D1, D7, D14, D25, D13. It decreases, germination also decreases in potentiated water in solution D4 and D12, D13. These results are also statistically significant. High correlation between optical properties of potentiated solution of sodium chloride and sucrose and their physiological activity was obtained. This suggested method can be used to predict physiological activity of potentiated solution. The results obtained really proved biological activity of potentiated water and process of potentiating. Thank you for attention and excuse for some problems with my demonstration. Thank you very much. Thank you, Dr. Manchanda.
Thank you very much, Dr. Maria. And I'm sure those little technical problems would not affect our enjoyment towards your presentation. So that is so our presentation for this section comes to an end, and now I would like to invite Dr. Erenthal to give his comments of the section and as a complete closing.
OK， 真係一個好非常之好嘅演講啊。OK。
Thank you for all the wonderful presentations.
關於一啲基礎啦、臨床前嘅研究啦 ，OK。
And it is really interesting to learn more about the basic, fundamental, and preclinical researches.
如果我哋咧係冇咗呢啲基礎研究嘅支持咧，咁順勢療法咧嘅臨床嗰個試驗咧，永遠都係企唔穩嘅。Okay.
And the clinical trials of homeopathy could never stand really strong unless we have enough the backup from these fundamental researches.
好高興咧睇到印度同俄羅斯咧正在進行緊咧好多不同類型嘅基礎研究。
And it is really our pleasure to see so many different kind of basic research ongoing in India and Russia.
第一位阿貝拉爾醫生啦 ，OK 即係有呢個即係喺呢個研討會入邊咧 ，OK 咁知道咧係不同嘅研究咧嘅人員啊，不同研究人員咧都努力咧開發不同嘅物理化學方法。
In this colloquium, we heard that many different groups are exploring different physiochemical methods for working out our black box.
大家都好努力咁去解構呢個未知嘅黑盒嘅，即係順勢療法喺醫學入邊嘅不同嘅假設咧，其實都好需要我哋咧去驗證嘅。
And there are different hypotheses in homeopathic medicines that has been developed and need to be investigated.
好高興貝拉醫生咧，佢嘅工作入邊咧，我哋咧可能咧，即係好高興聽到啦嚇。咁我哋我哋咧可能咧有更多工具咧嚟確保順勢療法呢個療劑嘅品質控制嘅。
And so it was really great to hear from Dr. Bellier's work that there may be more tools for us in the very near future to ensure the quality control of homeopathic remedies.
咁另外一位咧就考醫生啦 ，OK。
And then we goes into the presentation of Dr. Kroll.
喺好多唔同嘅國家咧，都係一個非常之重要嘅話題嚟嘅，即係 nosal 啲。
Nosal has been a really important topic in many different countries.
因為佢係一個咧係疾病嘅療劑啦。
Because of its nature as a diseased product.
咁但係咧。佢適當嘅，即係其實佢得適當嘅製備同埋利用咧，喺我哋嘅臨床實踐入邊咧係有好大嘅潛力嘅。
But its proper preparation and utilization could have a great potential in our clinical practice.
我哋好高興咧，聽到俾考醫生咧，佢講咧就係咩咧？就係即將出現嘅另一種新嘅瘧疾嘅 nosal。
So it was a great pleasure to hear about the new nosal that is coming ahead.
瘧疾喺世界好多地區咧都肯定咧，而係一種非常之重要。
Malaria is still a very important disease in many different parts of the world.
高品質嘅製作以及適當嘅標準咧，即係標準化對 nosal 嘅開發同至關重要，非常之感謝你哋嘅工作。
So preparation in good quality and proper standardization really is essential for the development. And thank you very much for your work, Dr. Paul.
另外咧，奧爾科夫醫生啦 ，OK， 好高興咧聽到咧係呢個水合富勒烯嘅資料嘅。
And Dr. Vyokov, it's great to hear about the new potential remedy of hydrated fluorine.
以後嚟自俄羅斯嘅研究咧，我哋能夠了解到咧另一種咧有前景嘅療劑，而且咧仲以配合晒咧係咁多仔細嘅即係喺呢個不同嘅模型入邊嘅證據嘅。
And we love to see researchers from Russia, and it is really exciting to learn about another potential new homeopathic remedy with such a detailed physiochemical investigation and biological investigation.
呢個真係令人好興奮。我相信咧有咁高標準嘅物理化學嘅研究咧，我哋喺不久將來咧，如果咧能夠咧以傳統嘅方法咧去驗證咧，會相信會十分之有趣。
I'm sure with such a high standard of physiochemical biological researches, if we can in our near future really prove the remedy with the traditional kind of proving method in homeopathy, it would be really, really interesting.
另外咧就係加瑪利亞啦，咁呢個另一個嘅嚟自俄羅斯嘅研究咧，有住即係有住同樣嘅價值啦。你哋嘅團隊咧嘅研究咧係有重要嘅發現嚟嘅，咁呢個咧係非常之即係。恭喜你哋嘅。
And Dr. Maria, congratulations! You've got another piece of beautiful work from Russia, and it is equally valuable. And your new discoveries are really exciting.
當中咧係最重要嘅係可複製性啦，即係呢個咧喺我哋嘅行業入邊，即係順勢療法行業入邊係非常之重要嘅。
And the most important thing is the reproducibility, which is really, really important in our profession.
我好真誠咁咧，希望咧喺世界上面咧更多嘅團隊咧喺同一個領域入邊咧，大家共同努力，大家相互學習。當然咧，呢個將會成為我哋所有人咧係創咗一個更加光明嘅未來嘅。
So we sincerely hope that more teams around the world would work together on the same ground, learning from each other, and surely it will pave a bright future for all of us.
好感謝各位工作同埋貢獻，多謝你哋。
Thank you so much for all your work and for all your beautiful presentation.
Thank you.
Thank you so much, everyone, for all those amazing insights, for showing us all those research works.
Just a minute, friend.
Yes.
So I would just like to give few comments about this session. This session is very unique because many new things have been discussed. First is about the malaria nosode. If we are able to take this to logical end, it can have wide, far-reaching public health implications for the world. And the second presentation of Dr. Jayesh Bellare can put homeopathy on the scientific pedestal forever. The moment we decide that we have to, it is a very strategic thinking that if we start working just below Avogadro, at least one major criticism of homeopathy gets a beating. But a lot of work is required because we have to prove further the differences between the biological activities and its clinical efficacy of these new potencies with the earlier, the way we are doing for the last 200 years. I really feel that at present, homeopathy is at a very, very turning point where we have to focus on one hand, the scientificity and scientific experiments to substantiate all our clinical successes that we have for the last so many years. So in respect of all the new remedies, even for the existing remedies, all such biological experiments are necessary. And this new way of potentization has potential to not only put homeopathy on the scientific pedestal forever, but has potential of increasing the success rate in clinical practice, because we can always be sure about the quality as well as we can measure the quantity of the substance with each potency. This will also help the regulators to control the quality uniformly all over the world. So such path-making researches are necessary in homeopathy. And Professor Vladimir, my congratulations to you, sir. Let us look forward this as a new remedy in homeopathy. And Maria, congratulations again. Lot of work is in Russia and lot of work is there in Russian language, which is not still known and fully known to homeopathic world. This is what I have realized during last 3-4 years with my interaction with Professor Vladimir and Russian colleagues. Especially, I'll be discussing during my presentation of significant molecules also after 2 sessions. So with these words, I'm extremely thankful to all the panelists who participated and gave wonderful presentations. And I would also like to invite all of you, if it is possible during the time frame, that on 12th of December, there will be a discussion on innovations in pharmaceuticals. And that will be in hall number 4, 11:30 to 13 CET, that is on Sunday. You all are invited to us as panelists, not as delegates, and we'll be sending you links also. So you all can join there and let us have further discussion what more needs to be done and what are the areas where a lot of innovations are required in homeopathic pharmaceuticals. Thank you very much for being here and sparing time for this. Thank you.
Thank you so much, Honorable Dr. Manchanda, for those very kind words. And thank you so much to all our chairpersons, our panelists who has shared their insights and research today. We just hope that these researchers would create a futuristic benchmark and inspire a lot of homeopaths throughout the world. Thank you so much for being here. Now, before we close the session, I would request everyone to please turn on your camera and put a big smile on your face so that we can have a group photograph together. Thank you.
Thank you.
Thank you. Thank you so much, everyone. Hope to see you in the next session, which is exactly in half an hour from now, which will be at 8 hours CET. Hope to see you all there. Till then, enjoy a great coffee break. And thank you so much for gracing your presence and making this session a very, very, very insightful and informative session. Thank you so much. Have a good day. Have a good coffee break.
Thank you. Thank you very much. Namaskar.
Thank you, dear. IB team, please note that put timer also for the next session. Along with the advertisement, put some timer on the screen so that whosoever visits hall, they know that now they have to join at what time. Okay? In terms of hours, in terms of minutes, that 20 minutes, 25 minutes.
Countdown timer. Put a countdown timer.
Put countdown timer. Oh, thank you.
Charlie, thank you.
Thank you for joining.